Okay, moving on from scientific method, we're now going to be moving on to some stuff with water. So why is water so important? Is because, well, it's the universal solvent. And you guys will be using those words like solvent and solution and solute within our next little formal lab that you guys will see that deals with chromatography. And basically what a solvent is that it's able to dissolve or break things down. So water is considered the universal solvent. Some things for you guys to think about. We're going to talk about why it's so fundamental for life, but it does make up about 71% of the Earth's surface, and about 60 to 85% of your cells is basically water. So if you think about like gooey, cytoplasmy, jelly-like material that holds all those organelles together, that is mostly water that we're going to see. Some forms that we can see it in three, some people call it four states, but the three exact states that we see. You can see it as a solid, you can see it as liquid, you can see it as gas. Some people say, well, plasma, but yeah, you don't really see it as a plasma, but solid, liquid, and gas are those three states that we can find water. So solid, just so that you guys are aware, is basically that frozen water, and it does freeze at about 32 degrees Fahrenheit, 0 degrees Celsius. Liquid water, it will boil at 212 degrees Fahrenheit, which is about 100 degrees Celsius. So why it's so important in this particular section is if we think about biology, biology is the study of living things. And theory-wise, if we go back to the beginning of life, life originated within water. So as our planet was first forming, very harsh conditions and lightning, we had no oxygen, we had lots of storms. Somewhere along the line, things started coming together, chemical reactions occurred. Theory-wise, we start to form organelles start forming little cells, and we get this single-celled life. So life actually originates in water. We just talked about cytoplasm and why it's so important within our cells, but also your blood plasma. So when you guys get blood taken at the doctors and they stick it in those red tubes and it's got the gooey stuff at the bottom, they then put them in a centrifuge and they spin it. And what they do is they pull all that solid stuff down to the bottom of the tube, and that liquid portion there, it's called your plasma. Well, if you're dehydrated and you have very little water or less water within your body, you have less of that plasma or liquid within it. And it's actually the plasma that they test. They're looking to see what your kidneys look like, maybe what your liver looks like, your different reactions within your body. They're actually testing the plasma, not the blood cells themselves. So you want to make sure that you're hydrated when you go to donate blood. And that's why they usually tell you make sure that you're drinking lots of water, because it is the water that is coming out because of all that cytoplasm, as well as blood plasma. About 3.5 billion years ago was our first cells. We actually have fossil evidence of these little micro cells, these teeny tiny little itty bitty bacteria. We have microfossils of them. So, structure of water. You guys should have done this in chemistry for all those you guys had chemistry, or first year bio also. The structure of water. So you guys should know that it is H2O. So what does that mean? It means that it's 2 molecules of hydrogen and 1 molecule of oxygen. Okay, so that's pretty simple. Yeah, H2O. Everybody kind of knows H2O. Let's go a little deeper. It says a polar covalent molecule. And that's usually where you guys go, I've heard those words before. Not really sure what that means. Polar covalent, whatever. Well, let's think about it. I'm going to kind of go off to another paper and look at some of these little molecules that you guys see here. So if this is my sheet, we know that oxygen, if you guys think back to chemistry, periodic table, it should have an 8, atomic number of 8. 8 protons, 8 electrons. Hydrogen, got 1. It's the first one on that periodic table. So if we were looking at that molecule of H2O, and we kind of sketch it out, I'm going to put oxygen in the middle. Remember, oxygen's got 2 electrons in that first ring. If this is not review for you and you're like, ooh, I'm not really sure what the heck she's saying right now, make sure you check with us so that we can go through this. We will look at basic chemistry really, really fast as we go through and get into organic chemistry. 2 electrons in the first ring, it's all it can hold, which means I have 6 more. So we have 6 in the outer ring. 1, 2, 3, 4, 5, 6. Oxygen is not stable. It wants 8 in the outer ring. So it's got 2 vacancies. So it is very unstable. We've got 2 hydrogens that are going to bond to that. If we look at our little hydrogens, I'm just going to put them as little H's here. It's got 1 electron in the first ring. That first ring wants 2 electrons. Hydrogen is also very unstable. So once again, what you should remember is that we are able to form what is known as a covalent bond. They are sharing electrons. Hydrogen's electron is filling oxygen's ring. Oxygen's electron is filling hydrogen's ring. So this is going back and forth at super fast speeds, and they are filling both rings at the same time. So because of that, hydrogen feels like he is stable. He's got 2 electrons, and this hydrogen feels like stable with 2. And now my oxygen, besides the 6 of its own, is kind of sharing this one 7, sharing this one, this is 8. This is not an ionic bond. Ionic bond means that I would physically give away or take those electrons in. I'm sharing them here. However, so we call it a covalent bond. But I said that water is a polar covalent molecule. So even though they're sharing oxygen, I say oxygen's kind of like this bigger bully, this bigger molecule. He's like, yeah, I'm going to share electrons, but I don't want to share them evenly with you. Because of that, he is going to pull more of the electrons to him. Because of that, it is going to slightly make oxygen slightly negative. It doesn't mean that we're giving or taking electrons. My hydrogen, since that oxygen's pulling more of that electron, not really sharing it evenly, is going to have a slight positive charge. So that right there is a single water molecule. H2, my 2 hydrogens, O. Single water molecule. It is a polar covalent molecule. Means that we are not sharing the electrons evenly. But it is not an ionic bond. It's still covalent. They're still sharing. They're just not doing it evenly. So now I can say, well, if that one water molecule bonds to another water molecule, let's say here's another one down here. He might look like that. Maybe I got another one over here. One looks like that. They can actually start sharing together as well. And we're going to see that in a moment. So I'm going to track back to this one here in a second. But you will always see water kind of in this fashion here. So we say it kind of looks like, if you want to draw it out like that, it could look like little Mickey Mouse ears, and you guys will see that on one of the worksheets that you do. So here are the hydrogens. Here's my oxygen. You will never see it like this, a linear molecule. That would be a non-polar covalent molecule. That means they're sharing them all evenly. We're all nice and even. That will never happen. Because oxygen is pulling more of those electrons, those hydrogens kind of get pulled closer together there because of the pull of those electrons. So that is your polar covalent molecule of a single water molecule. We'll look at various water molecules here in a moment. So going back, we just said, basically what I just did is what you guys are going to see here. So charges are distributed unequally. Oxygen's got a negative charge. Hydrogen has a slight positive charge. These are known as dipoles. Okay, that positive and negative regions. It's because of that dipole, it's because of those positive and negative regions that it is able to hydrogen bond with other polar molecules. So a single water molecule is a covalent bond, sharing electrons. But if I join this water molecule with this water molecule, it is going to hydrogen bond. So hydrogen bonds are now going to form between the hydrogen, which has a slight positive to it, of one polar molecule to the oxygen of another polar molecule. Opposites attract, positives and negatives. They're going to attract to one another. So here we go. We are doing this. And I drew a few more. Let me give you another one over here. It might look something like that. Water molecule, water molecule, water molecule, water molecule. What's going to happen is this guy is slightly positive. Oxygen slightly negative. Opposites of my water molecules will attract to one another. So it's kind of like magnetism, and they will be attracted. That is a hydrogen bond. These bonds right here are covalent bonds within a single water molecule. But when I join separate water molecules, my positive and negative opposite charges are attracted to one another. So once again, this hydrogen can attract to this negative oxygen. He's positive, he's negative. That is now my hydrogen bond. So hydrogen bonds are going to form between various water molecules. It is a super weak bond. It is easily broken, that particular bond that you guys see there, okay? And that's what we're looking at in this last little bit of this notes on this particular page. So they are made and broken easily, and that polarity allows us to dissolve many substances. And why it's so important within the body, because we're able to utilize it within blood, and we have different reactions, and things can be broken down, and that's where we're going to see it. We're going to be talking about some other words, but words that you guys should know. Break words down. Science is vocab. Know it. Hydrophilic, hydrophobic. Know the word hydro. Hydro means water. If you have a phobia of something, you have a fear of something. Arachnophobia, a fear of spiders. So something that is hydrophobic means that you have a fear of water. Hydrophilic means water loving. So for instance, polar molecules are attracted to water. I can put something like sugar. You go home, you decide you're going to make some Kool-Aid, you put some sugar and some water, a little bit of flavoring. That sugar is going to dissolve because it is hydrophilic. It loves to be near water. If you put oil within that water, that is hydrophobic. It wants nothing to do with it. It doesn't want to blend with it. It doesn't want to mix with it. It's not going to break down. It is insoluble in water. So we can say that non-polar molecules that repel water are hydrophobic. So for example, oil is hydrophobic. It does not dissolve in water. It is said to be insoluble. Because of the hydrogen bonds that we just created between various water molecules, certain properties are going to exist. So we can say hydrogen bonds result in properties of water, such as what is known as surface tension. And we say that water has a very high surface tension. All those little water molecules are sticking together. Within surface tension, and these are the words that you guys will be utilizing within your chromatography lab, we have things like cohesion and adhesion. Words you should have heard in first year bio. You're probably like, yep, heard them, have no idea what they mean. Here we go. Let's reiterate what they mean. Cohesion means the ability to resist the breaking of hydrogen bonds under tension. So you're going, all right, so what does that mean? Well, basically, water molecules are sticking to other water molecules. Cohesion. All these water molecules are sticking together. So if you look at a bug that's walking across water and you go, oh, look at that bug. It's really cool. It's kind of walking across water. How come I can't do that? That bug is light enough that he is not breaking the cohesion. He is not breaking those hydrogen bonds between water molecules. You guys try to do it. You got a little more mass than that bug does there, and you're going to break those hydrogen bonds and go right through water. Adhesion is the ability to stick to other polar molecules. So the attraction of unlike. So for instance, water to glass, first year bio. If you guys ever had those microscope slides and they're like, oh, wash them, but lay them out flat, don't stack them on top of one another. Because what happens if you put microscope slides on top of one another, glass on top of glass, and let's say there's a little bit of water in between, they actually will stick together, and that water between it is adhesion. It adheses it together, sticks it together. I can also think of adhesion and cohesion as the way that molecules are able to move up a tree. So let's say that here's our little tree. I'm not the artist, so go with me here. Okay, so what happens is we need to get water up to the top of the tree there. So how is that water going to get to the top? Well, we can use adhesion. And what happens is after it rains, water goes in and it gets picked up into the little roots. Going to move it up through the xylem, which is the little transport system within the tree, and those little water molecules are going to stick together. Cohesion. And as they stick together, they're going to force the molecules to move up the tree. I can also see adhesion, because I just said we have the xylem, which is the transport tissue within the particular trees that we're going to see. Those little water molecules are also going to stick to the xylem and move up as well. So they can stick to each other, cohesion. They can stick to unlike substances, which would be the xylem, so water sticking to the transport tissues, and that would be your adhesion, moving up. The way things move up against gravity is what is known as capillary action. So it's the movement of water, or any liquid for that matter, along a surface caused by cohesion and adhesion. It moves against gravity. You are going to see this hands-on with our chromatography lab that we will be doing. but basically think like paper towels. If you have a spill on the table and you take your paper towel, maybe you go like this with it, that water is going to start transporting itself up. That paper towel sucks up the water. It's also the way that plant roots are able to pull water up the tree, like we just demonstrated right there. In addition, what we have there is drinking up. So it's basically the capillary action of water molecules into a substance and swells. If anybody planted seeds, I know gardening was huge this past summer because everybody was home and they wanted to do things outside with their kids. So maybe they planted a garden and maybe you planted some seeds. Seeds go through this action. You're putting them in the ground, you're watering the ground. Well, that water gets sucked up into the seed and it causes seed germination. And the reason it germinates and we get this seed that actually is going to swell and burst is because the water is moving into that particular seed. Eventually those little rootlets, the little roots that come out of it. And if you've ever seen or gone to like a pond or a lake and you pull this old piece of wood, maybe there's a piece of wood on the bottom, you pull it out and it's super heavy, it's waterlogged. In addition, we have swelling of it. Water has moved into it. Also absorbent towels. So maybe when you get out of the shower and you're drying your hair, you're drying your hair with that towel, it's pulling the water from your hair. So it's kind of like helping it to swell. That towel is pulling it out there. Okay, so those are some properties that we have of our water. As we move on, some other different things that you guys are going to realize. We talked about specific heat and things like that. So we know that water is going to have resistance to temperature change. It's going to have a high increase or high chance of resistance to temperature change. Meaning it takes a lot of heat to change the temperature of water, which is really, really good. Think about hot summers. That hot air, 100 degrees, if our ponds heated up as quickly as our air temperature did, that water would pretty much be too hot for organisms to live. So in order for things to raise or change temperature, we have this high specific heat. You may have done this in chemistry lab. You know, how much heat does it take to raise 1 milliliter of water 1 degree Celsius? And it takes a lot of energy to do that as opposed to maybe other liquids like alcohols and whatnot. We also have a high melting point. Things for you guys to remember. In order to form ice, water is most dense at 4 degrees Celsius. Okay, water is most dense at 4 degrees Celsius. Ice is 10% less than 4%, which is why ice floats. Because if that water started freezing from the bottom and worked our way up, think about all our little critters there. So if water froze from the bottom and worked its way up, all those fish, organisms, living things would be up on the surface and eventually would die. By having that 4 degree water up at the top, having that frozen water at the top and working our way down, that 4 degree water allows organisms to survive down there. So it kind of insulates it. Ice comes from the top and works its way down and gives them kind of this little layer to insulate and allow them to survive. So my ice forms at the top, my 4 degrees Celsius is most dense, and that's not going to freeze, but it's going to work its way down so that organisms are able to live. So super important for living things. Most energy from heat is used to break those hydrogen bonds instead of causing the molecule to move. And that's why we have this high heat of vaporization, high specific heat, high melting point, high boiling point, as well as everything else. Chromatography lab, like I said, I keep bouncing back to this. You guys are going to see words like this: solvent, solution, solute, things that you should have seen before, heard before. So here we go. We said water is a universal solvent. That is the liquid that does the dissolving. Can other things besides water be a solvent? Absolutely. First year bio maybe used hydrogen peroxide instead of water as a solvent. Anything can be used as a solvent, liquid, as long as it is breaking things down or dissolving things. That solvent is a large part of the solution. Solution is my solvent and my solute. The solute is what the solvent is breaking down. So you go home, you're making iced tea mix with water. Say that iced tea mix is going to be your solute, water is going to be the solvent. You put them together, you stir them up, you mix them up, you just made a solution there. Let's see, usually your solute is going to be lesser quality. Solvents, what we can dissolve ionic compounds, NaCl, okay, salt water, or basically salt. We can create salt water. We take water, put it in there, we're breaking the bond between sodium and chloride. We can dissolve polar covalent compounds such as amino acids, and we'll get into amino acids when we look at our organic chemistry a little bit more. We can also do smaller organic molecules such as sugars. So carbohydrates are larger organic molecules. Sugars are the smaller version of that. Once again, not only amino acids, but we'll also look at sugars when we get to organic. Acids, bases, and buffers, hopefully touched upon this first year bio as well as within your chemistry class, because it's those hydrogen ions that we see within that, those H pluses, which means that we've kind of given away electrons there that affect cell structure and function. So it's not like acids and bases, chemicals that you think of, but it happens within our body all the time. pH plays an important role within our body, and we have different pHs within our system. So our blood will have a certain pH, urine will have a certain pH, your stomach will have a certain pH, your kidneys function at a certain pH, and they can all be different. It's not like you just have one standard pH all throughout your body. And if that pH is off within that system, it can actually lead to death, because enzymes, which are certain special types of proteins that help speed up reactions, are dependent upon these particular pHs, and they can denature or get broken down very easily if the pH is not correct. If those enzymes don't work and reactions don't occur, it can actually lead to death. So water can break apart into hydrogen and hydroxide ions. So these H pluses are my hydrogen ions, those OH negatives are my hydroxide ions. Okay, so because of that, if we look at water, H2O, water is neutral. We have the same number of hydrogen, oxygen, positives, negatives that we have within there. So it is going to be neutral. We don't have more of hydrogen ions or less of hydrogen ions. And we'll see that in the pH scale coming up for you guys. Remember that pH, when we talk about pH and what it does, it actually is going to go through and measure the amount of hydrogen ions in solution. The more hydrogen ions you have, the more acidic something is. So if I have equal amounts of hydrogen and hydroxide ions, it's neutral. It's a pH of 7. But if I have more hydrogen ions, I'm going to become more acidic. 7 to a 6 to a 5 to a 4, and so on. If I have more hydroxide ions, which means I'm going to have less hydrogen ions, I'm going to become more basic, and that's anything higher than 7. So 7 to 8, I have more hydroxide ions. 8 to 9, I have a lot more hydroxide ions. And things to remember is that bases can be just as dangerous as your acids. So you're going, well, an acid of 6 to 5, well, that's not that bad. It's only 1 over on the pH scale. It's actually a lot more dangerous than what you guys realize. So thinking back, pH scale is measured from a range of 0 to 14. We'll see another example of this. I want to stay on the next page. 0 being the most acidic and 14 being the most basic, 7 in the middle, smack dab neutral. And what that means, once again, is that my hydrogen ions are going to equal my hydroxide ions. I have no increase of either one, which is why water, H2O, is neutral. I don't have more hydrogen or hydroxide ions. They are equal there. Every time that we move along that pH scale, remember that it is going to kind of be logarithmic. So it is 10 times stronger. It has 10 times more of those hydrogen ions in solution than the previous number. So you can be like, oh, I've got a pH of 6. It's not that bad, as opposed to a pH of 5. Well, it's 10 times more. So 7 is neutral. A pH of 6 means you have 10 times more hydrogen ions. pH of 5 means I have 100 times more than what I had at 7. I have 1,000 times more for a pH of 4 than what I had at 7. And the same goes the other way, but now we're dealing with those hydroxide ions. So 10 times as many, 100 times as many, 1,000 times as many, and so on. So each change of the pH is 10 times stronger. You just can't say, oh, it's one step over on the pH scale. It's just 1 time stronger. It's 10 times stronger than the previous step that you have there. Okay. Acids and bases. Acids, all substances that donate hydrogen ions. We said that there are more hydrogen ions in solution. Your bases are the ones that are alkaline. They have more hydroxide ions within them. You guys somewhere along the line may have used different ways to calculate out pH of things. And there are multiple ways to do it. Probably the most accurate way, which means that you're going to get quantitative data. The most accurate quantitative data is going to be a pH meter. You stick a little probe, you put it into your solution, it gives you an exact number. Actually, it'll give you down to decimal point number, telling you how many hydrogen ions or hydroxide ions do we have in solution. You might say 7.4. It's great if you're checking see something that you think is neutral, but we want to make sure, is it slightly acidic, is it slightly basic? We need to know a little bit more. So that pH meter gives us the most accurate information. We also have litmus paper. Now, litmus paper gives you more qualitative analysis, meaning it's not really going to tell you too much more. It's going to let you know, are you dealing with an acid? Are you dealing with a base? Are you dealing with something that's neutral? So it's going to be more descriptive rather than something that's quantitative, giving you a number on that particular pH scale. Litmus paper, you have red paper, you have blue paper. You have one of each. You have an unknown solution. You're going to take that litmus paper. You take the blue, you dip it in. You take the red, you dip it in. If red stays red and blue turns red, then you're dealing with an acid. So anything that is red is an acid. My next solution, dip red and blue. You always have to test both. Dip red in, dip blue in. Let's say blue stays blue, red paper turned blue. It's because of the reaction with the hydrogen and hydroxide ions. Anything that blue is going to be basic. If you dip them both in and neither one changes, it means that I have a neutral solution. The one that you're probably used to using most of, if you have a swimming pool or you have maybe a fish tank and you check the pH of that to make sure that that environment is healthy for the fish, you're probably going to use pea hydrant paper. It's orange paper. You pull out a big piece, you dip it into your solution, and it's going to change based on the color wheel that you have. So once again, red is going to be your acids. It may go pink, it'll start to go green, it will start to go blue. By the time you get down to blue, you now are going to be within that base range. So it's going to give you more of that qualitative as well as quantitative. It changes colors and it gives you a number. It's not going to be exact. You can't go, oh, look, there's a 7.2 with the pea hydrant paper. No, you can say maybe 6.5. You can maybe go between two of them. But the solid numbers, you could say it's definitely a 6 or a 7 or an 8. It's going to be better for that. You can also use indicators that will also describe. So red cabbage juice, for instance, if you boil red cabbage, put it in some water, it can actually make its own little color wheel so that you can determine maybe unknowns. So if you don't have any of this stuff, there are ways that you are able to do that, and it's more of a qualitative or descriptive with that. Not too much of strength. It won't really give you too much number-wise, but it will let you know whether you're dealing with acids or bases, and it's a way that you could do it if you didn't have any of these other methods there. I'm going to skip over to this side first because there's my little pH table that we were just kind of working with. So it says when a strong acid and a strong base interact, a salt is formed. So if I take that strong acid, HCl, hydrochloric acid, and sodium hydroxide, acid, base, I join them together. We have this reaction. It's called a neutralization reaction. And when this occurs, my hydrogens as well as my hydroxides are going to kind of combine together, and it creates water, and it will also create some salt for us. So you end up with NaCl and you end up with some water there. So equal things of equal amounts. So if I put them together, I can get something that's more of a neutral solution, which is why it's neutralization. Here's my scale, 0 to 14. 7 smack dab in the middle is neutral. Pure water is neutral. Anything less than 7 is going to be an acid. Anything higher than 7 is going to be a base. The farther away you go, the stronger it becomes. Some characteristics of an acid: it is usually sour tasting. If you think about things, lemons, for instance, they're usually sticky, they're sour. Some examples of things that you guys have: stomach acid, lemon juice, pH of 2, lots of hydrogen ions. Soda, vinegar, if you are a soda drinker, you're probably wearing some of the enamel off your teeth because it has a pH of 3. Coffee is a pH of 5. Urine that your body produces has a pH of 6, so just slightly acidic. Your bases are usually going to be more bitter tasting, is more slippery if you put on your finger. Things like cleaning supplies, like soap and substances like that, they're usually more basic. Your saliva, seawater, pH of 8. Milk of magnesia, people take it to kind of calm their stomach acid. Well, we're taking a kind of a stronger base, stronger acid, combining them. We're trying to neutralize it just a little bit. Ammonia and bleach are your strong bases that you have there. Buffers. My last little bit before we look just real quick at some stuff with reactions. So within your body, you have something called buffers, and buffers basically help maintain the pH within your system. We'll also deal with buffers when we get to gel electrophoresis. It's going to maintain the stability of the gel that we're putting DNA or amino acids into so that we're able to run it. Depends upon what the pH that we want to maintain it. For gel electrophoresis, we want 8.2 to 8.6. If it becomes too acidic or too basic, it's going to destroy the substances, the DNA, the proteins that we may have within that particular gel. Buffers within your body maintain the pHs within those systems so that they are able to run correctly. Your lungs and kidneys, remember, they filter waste. They balance the pH in your body. They are exhaling out carbon dioxide. If you keep that carbon dioxide within your system, it becomes too acidic. So we're releasing hydrogen waste. It's helping to maintain homeostasis and internal balance. If you start to build up these different things and your buffers are not working, you're starting to build up hydrogen ions, you can go through something called acidosis. Too many hydrogen ions are present, so kidneys and lungs aren't working. We build up hydrogen waste. A lot of times we see it with diabetics, and it can actually lead to coma. It can actually lead to death. If we go the opposite way and now we are too basic, what we can have, usually we see this with pregnancy, sometimes we have this buildup of hydroxide ions within our system. It can lead to something that is toxic, can lead to convulsions, it can lead to problems within the nervous system. So what buffers are, you have bicarbonate salts. They're used for your primary buffering agent within the body. Bicarbonate salts help to maintain homeostasis, internal balance. My blood should be close to neutral, about 7.2, 7.4. So I want to make sure that I maintain that. Your body has these systems, these buffers that are allowing us to do that. Okay. On the back, just real quick, you guys are going to go through this on your own, but it just goes through some chemical reactions. Look at them. Let me know what your questions are on there. These are ones that you guys should have done in chemistry. Go through them. We also went through them in first year of bio, so whether we're talking about synthesis, building up reactions, remember these words: dehydration, synthesis, hydrolysis, building up, breaking down. We are coming back to these again, okay? Down the bottom, it talks a little bit about coefficients, subscripts, and how to balance equations. It is important. Am I going to put them on tests for you guys and say, oh, let's balance these equations? No, but you need to know that things cannot be created nor destroyed. Whatever goes in, I need the same amount coming out. So if I have H2O, this coefficient in front means I have 2 molecules of water. So I am going to have 4 hydrogens because I'm multiplying them through, and I have 2 oxygens. I need the same amount on both sides. Okay, so if I have a superscript, meaning I have 2 of these CO2s, you'll see that above. But it's important to balance equations. You'll see it more in chemistry. You'll see it more in organic chemistry. Okay, so sorry about the long note taking there, but hopefully that helps explain some things for you guys. If you are not sure and this went a little too fast for you, come see us. This stuff should be reviewed, but it's important information that you guys should know. Some of the other things that you guys are going to see on this particular tab with your notes, there's a whole sheet that's going to explain why water is so important within the body. From digestion. If you're dehydrated, things are not digesting through your body as quickly. You can't absorb nutrients as well. Lubricate your joints. So maybe your knees are a little sore. You don't have that synovial fluid there between them. Maybe you're dehydrated. It flushes your body of waste material. When you sweat, it's mostly water. So if you are overheating and dehydrated and you are not able to sweat, it means that you do not have enough water within your system. And that's not a good thing because it doesn't allow your body to cool down and get rid of waste material. So that's where that regulating of body temperature comes in. It acts as a shock absorber for the brain as well as the spinal cord. It converts food components. It helps break down digestion so that you're able to absorb nutrients. Go through this sheet. It talks about properties of water, surface tension, as well as other things, and how your body kind of changes as you age with the amount of water that certain parts of your body may have. Okay, there's also another little sheet on there, and I don't know if I have it in front of me. There's a little sheet that talks about properties of water as well. It looks like this. This is just an FYI kind of reference sheet for you guys. It goes through the dipole of water molecules. It goes through its properties once again. It talks about pH on there, talks about some other little things that can get dissolved. And this may help you with your water questions that you guys will see that will be assigned to you guys eventually. So once again, this should be review. If any part of it was fast for you, go back, rewind, slow it down, come see us, ask for help on this. This is the building block that we're going to start building upon for some other things. So if anytime something goes too fast, please, please, please make sure you come and get some help. Thanks.